commitments are there. So we should have work-life balance.
Okay, Dr. Chandri. So, with due respect, after hearing from you, we will send an official mail to Sudhira, and also if Parvati is not responding, we will be approaching her
I
via
think
mail.
Parvati is there. If
No,
she's
just want to confirm
if it's
because
in a long leave, we can send mail to Parvati too. But
Yeah,
this
sure.
way we can send to only to Sudhira.
Okay, Chandri. Yeah.
Sure.
I think Nikhil and Prashant also agrees with this. Since there is no contribution from Sudhira for a long time, we will be sending her an official mail and confirming her contribution.
Um, okay.
Yes,
So,
Nikhil, you want to say anything on this?
the no, I was going to talk about the progress.
Okay, great. Go ahead.
Okay, so the progress is that we realized that the previously selected structures were mutated structures, and that, like, if you look at the WHO sheet, um, it says here that we need specific haplotypes or specific strains of the disease. So, like for PFCRT, we had 6 UKJ, but that did not align with the haplotype we needed to have. So what ended up was we had the wrong wild type. So now we've reselected all of the proteins. For PFCRT, we've had to use Swiss Modeler since there was no PDB structure for that strain of the disease. We've used Swiss Modeler for these 3, and for these we've swapped PDBs, like changed the PDB we're using.
Okay. So we have to remove this IDs, and we can officially claim that we have done modeling. Correct?
No, sir. So for these ones, these are correct IDs now.
Okay. So
These
which
ones
one?
are the
Which one
correct
are
wild
modeling?
types. These 3 are Swiss model.
Good. Oh, okay, you have mentioned already. Fine. So no need
Yeah.
to mention like structure from Sys modeler. That is understood. Like you can write it. Fine. It's enough to
Yeah.
give preprot ID.
Okay.
Yeah.
We only have to redo about 3, 4.
So how
And
out of 7, how many 7, right? Out of 7,
Yeah.
how many models and how many experimentally determined structures?
there are 1, 2, 3. There's 3 Swiss model and 4 experimentally derived.
Okay, 4 experimentally derived. Fine.
Yeah.
Got it.
Also, so I don't know if we've brought this up already, but I had found a little bit of a discrepancy in the blind docking. Should I share that with you? Has that already
Okay,
been shared?
please.
Okay, wait. I'll just bring it up, sir.
See,
I think,
yeah. See, all your hard work have to be displayed and we can cross check, right? Even though it's not helping us in a mainstream, somewhere definitely we'll try to fit in, or put as a supplementary file. But I kindly request, you guys are not doing that, but as a general instruction, never manipulate the results. Show what is present.
Yes, sir.
Yeah.
Okay, yeah, found it. These are just, I don't have it on the PPT. These are just messages I sent to doctor to cross-check. So
Yeah.
I'll explain. The issue was that whenever we were performing blind docking, at least for me, the ligand was binding in the wrong site. So I figured out which site it binds in from experimentally derived structures. So
Okay.
PFDHFR has, like, I figured out where the binding site actually was. Then I did blind docking, and
Okay.
for blind docking, it binds in this crevice. You can see on the orange image.
Okay.
Purple image, it binds on a different crevice, which is the actual. Oh no, sorry. Orange image is the binding site. This is the correct one.
Correct one in the
And
sense where the mutation have happened, correct?
Yeah, it's the actual, like, experimentally derived binding site.
So this is, you've made a site specific or you just gave randomly? It's
This is
random.
site specific for the orange one.
Okay.
And the purple one is blind docking, where I just give the whole protein space and let
Fine.
it bind.
So whether it picked it properly or it is binding somewhere else.
It picked it incorrectly. You can see on the yellow image.
Okay. Yeah.
This
Now,
one is the binding pocket in the bottom, and the purple one is the blind docking one.
Yeah.
So
So
just because, yeah, so the blind docking might be binding to incorrect sites, and it's not like the effect of the mutations isn't coming in as a result.
fine. So may I put in this way? Have you heard of allosteric inhibition and competitive inhibition?
No, sir.
Can you
Yes,
please type it? Just you can type it.
Type what?
Allosteric. ALLO
Allosteric
and competitive inhibition.
and
Competitive inhibition. Yes. Go to images, please. Yeah, the second image works. So what you guys can understand from this?
inhibitor
I can't-
competitively bind in the place of the medicine or drugs.
Correct. Okay. For example, in your case, malarial protein is there. Fine. Or let me put on this. On enzyme A is the lock. Clear? Yeah, fine. Don't change here. Just look at that enzyme is the yeah.
Okay,
Fine.
okay, I'll go back to this
See,
one.
both are same, but yeah.
Yeah.
Can you get the blue color bulb is a lock. Okay. And substrate is the key. Substrate is
I
what?
I
What
think
if yeah, what if the lock is getting open? Then we can say it's a danger. The disease may occur. What is the inhibitor is doing in the first case, the blue blob? What it is doing?
it's deforming the active site so the substrate can't bind.
Yes. So it's blocking the substrate. It is competitively binding because the conformation is very flexible, suitable for that particular inhibitor. Clear?
Yeah.
Whereas it's one way of inhibition. What about the second one? Second
Sorry.
one is allosteric inhibition. Even yes,
Bind to the allosteric site.
yeah, bind to some other site, but even though it can change the conformation of the particular binding plot. Clear?
Yes, yes.
So you're saying that the it's binding to a different site, but that could be allosteric inhibition, so it'll change
Ah.
the binding site anyways.
Yeah, we are not
Okay.
sure about it. We are not sure about it. So we have to check both the results.
I'm
See,
pretty sure the PFTHFR I got,
Ah.
it was bound to the same ligand, so like the binding site should be correct for that inhibitor.
Correct. So either you can select the grid properly or give the string in the binding sites, and that is a kind of blind docking. Sorry, that's a kind of site-specific docking. And we also cross-check with the blind docking whether it is allosteric is happening. Okay. So since the
So-
protein is so big, it can go and bind anywhere. But luckily, your allosteric, okay, luckily in the blind docking also, if your ligand is fair enough, your software is able to detect that particular locus of your ligand binding active sites, then it is great. So I appreciate the efforts. You can show both the results.
So should we continue with blind docking or
Both.
site specific?
Both.
Oh,
Bring the
okay.
results. I think it's easy only, I guess. You have how many combinations?
we have as it combinations as in like that.
7 into? I think doctor can answer. Doctor, how many combinations we have?
I think
Dr. Vijay?
combinations like this. For wild type only, sir.
Yeah, for only wild type.
For wild type, there's 1 to 10.
Okay, for mutants altogether?
Altogether, there's 12.
Around 66, sir. 66.
Yeah,
Yeah,
66.
66. So 66 you can do both blind and comparative. We can check the way.
Okay, sir.
How many
Oh.
is completed so far?
How many?
Is completed so far? I think you guys might have completed at least blind or site specific by now.
So we've completed blind docking for we had to just change the proteins now, sir, right? So we've completed blind docking for these.
Yeah.
The 3 that were unchanged. Sorry,
Okay.
the 4 that were unchanged.
Yeah.
And for the other 3, we'll have to redo.
Sorry.
Nikhil,
So in this
uh,
1
you,
week,
you have
yes,
you
yes,
have
doctor.
completed the Nikhil. Please explain how to complete. How you can use the programming or manual docking. Please explain that part, because we are
Okay.
using the manual already learned from the Nyberman internship program, but you use some different way, I think.
Okay. So the way I was completing blind docking was basically, since blind docking, you don't need to find the site, and it just has to encompass the protein and have like that. So I made a script.
Yeah.
I think I can show it to you, but should I
So,
show it to you?
Mr. Nikhil, one thing we have to clarify. Can you
Yeah.
compare a marathon runner with an athlete, 100-meter dash sprinter? Is it fine or fair enough?
No.
Both are runners, both are athletes, but what a 100-meter sprinter will do and what a marathon runner will do. Are they sharing the same platform?
No.
Same way, either all your team members have to follow your script, or you have to follow their way. Then only we can have a standard comparison. Getting the point?
Yeah. So okay.
I really appreciate, man. This age, you are putting a lot of efforts. You are trying to explore new things. That is well appreciated, much appreciated. But for the study, we have to give a common methodology. Fine.
Okay, but
You can convince your team, like
yeah.
for a site, if you're making a smart pay, you can convince your team, and if it is even smarter, you can do all the stuffs in a single stretch and show them. If you're getting a better results, definitely we can include that. Fine.
Okay, I think that's something we could do is for all the blind docking results, maybe we could use a program since you don't need to define the binding box, and for
And
the site specific, we can do it manually if that's
yeah,
fine.
let me cross check now. And Dr. Vijayanta, what about the progress? Like blind docking is done, or it's site specific is done?
Sir, we have already done the blind docking part.
Okay. Okay, fine. Nikhil, actually, for this time, you try to find the site specific docking using your program. Is there any way? Try to find that way.
Okay, sir.
And try to choose the parameters same like our Autodoc Vina.
Yeah, so it's using Vina only.
Yeah.
It's
If
the
you're
same
using
software.
same software, if you are using Autodoc Vina, then it's fine. And if you are able enough to replicate the results, like blind docking results for all the complexes, all the 66, that's well and good. You try, give
So
a
I
try,
can do that.
but don't disturb the results over here. You also
Okay.
have to contribute for your part, like what others have did.
Okay.
You're getting my point? Take that as a priority. Give the results to Dr. Vijayanta so that he will include your data into the results. And along with that, you can try in your own programming part. Fine.
Okay, sir.
I'm happy that if it is Autodoc Vina is working out good, and you can sort it out that site specific too, then we can go with your flow. But meanwhile, Dr. Vijayanta is trying to get the meaning out of the results the traditional, conventional way. Conventional way in a sense, with step-by-step protocol. Okay.
Yes, sir.
Okay.
Yes, sir. Yes.
So first, Nikhil help them, give your part, then you follow this. I think 1 week time is there. We can easily sort it out.
Yeah, sir.
Yes, sir.
Hope I'm making sense. And sorry for continuous delay. I'll once it's done now, we'll look over the methodology part and writing part. Clear? And then I'll teach you what. See, the table is our major hold here. Then I will teach you how to make better visualizations. And I think you have good sense of making a visualized graph. But there are few other interactions have to be analyzed. That will teach you, and we will generate some special plots like violin plots, KDE plots. Fine. These stuffs we will discuss in the near future. Might be the next week and 1 week later, 2 weeks later. Clear? So without further delay, you are
Sir.
going at a good speed. Try to rush up the process. Yes, doctor.
Sir,
Yeah, please.
last week you have give me some points to do the homework, like number
Okay.
of interactions with histogram or
Yes,
diagram.
yes,
But
yes, yes.
I have no idea, sir. Have you any pictures or photographs?
Number of interactions.
Interactions with histogram and diagram.
Just a minute. I'll
How
show
to
the
prepare, sir?
manuscripts.
Okay, sir.
Yeah,
Okay.
please. Have we discussed, since we are doing protein ligand, do you discuss PLIP? You all remember PLIP session
Yes, sir.
files
Yes, sir. Yes, sir. Yes.
and Discovery Studio? You all remember?
Yes, sir. Interaction photographs like 2D, 3D.
Yeah. Yes, yes, yes.
Okay,
But before
sir.
the same thing only I'm asking. Before proceeding to that, let me see the table, consolidated table.
Okay, sir. That
Clear?
was already prepared for the manual docking part, and
Yes. Good. So get the results of Nikhil's too. Let Nikhil do in the same conventional way. Show the result all together, and let Nikhil perform all thing in his own way, like own way in a sense with the automated way, Python
okay,
way,
okay,
and we can come.
okay, sir.
Okay.
Then we decided
It's-
which will be the
Yeah, which is having more counts, which is showing
okay,
more
okay,
positive.
okay,
That results we can retain.
okay.
Yeah. And Nikhil, be strong because you should not waste your time. I think you should go in a proper direction. Okay.
Okay, sir.
Fine.
I
And yes.
there just might be like a little slowdown because my boards are coming up, but
Oh,
I'll try to do work in between. My boards
Yeah.
are in like 2 weeks, so
Then
Okay,
meanwhile,
nothing.
what you can do, you can do the conventional part and give to Vijayanta, and later on for specific you can try your way because at least one part have to be completed. Okay.
okay, sir.
Yeah. So
Okay,
please
sir. Okay,
do
sir.
your work and give, and let us proceed. And thank you so much, guys. You guys, we can meet in the next class. And meanwhile, next Monday. Meanwhile, you share the results in the group, and you can contact me in the group. You can tag my name so that I can respond you.
Sir,
Okay, sir.
Yes,
sir,
yes,
another
doctor.
another question is, you have said in the last class, understanding mechanism of the artemisinin, chloroquine, and resistance mechanism. I have
Yes.
learned several articles related to this, but how to
Fantastic.
how to prepare the file for the article, like discussion paragraph, like short of paragraph, like
Okay.
file I have to prepare.
Yeah, like see, you want to convert those statements in a manuscript,
Okay, like discussion part.
like for discussion part. Yeah, for discussion part, even in introduction part, like we have to fill the research
Okay.
gaps, right? We have to find the research gaps.
Okay,
By writing in that way, you can very surely say how far our study might be significant. Correct?
Yes.
See, as a researcher in this malaria field, you might know how important our study is. Correct?
Yes, very important, sir.
Yeah. So that importance you have to reflect there. Clear?
Yes.
Oh, okay. Thank you so much, team.
Okay,
If
sir.
any
Thank you,
doubts,
sir.
any clarifications, yeah, because next
Thank
team
you,
is
sir.
waiting. Thank you, Nikhil, and thank you, Dr. Vijayanta. Fine.
Thank you, sir.
We'll